Perfect. Yeah, I also got confirmation. Hello, everybody, one more time. And I'm Raghavendran. I'm a research consultant and mentor with Pine Biotech and OmicsLogic. I'm trained in bioinformatics and I hold a PhD in computational biology from Max Planck Institute, Germany. My broad research interests include are in the application of data science methods to understand cancer biology. My role in Pine Biotech includes developing curricula for the platform and also mentoring students in the research fellowship program and also in the programs that you'll go to sign up. Please allow me to talk about few example projects that we have prepared in the platform Learn OmicsLogic platform that will help you appreciate how bioinformatics analysis can be leveraged to understand the biology of pathogens and to study infectious diseases. So first, we will focus on a project that studies the current pandemic, COVID-19. So in this project, we explored the pathogen Sars-Cov coronavirus itself and as well as few diseases that are caused by it. So this project will help you explore the origin of the pandemic and how different diseases are caused by this pathogen and how they are related to existing strains from human coronavirus and also to other strains from animals sources also. So we will do this by using publicly available data from repositories like NCBI virus and GEO data, etc. So we will learn in this project, we will learn to perform or how to perform multiple sequence alignment followed by phylogenetic analysis. These analyses will help us compare the existing strains of coronavirus to understand the zoonotic origins of the disease. So we will also learn in detail how to interpret results from analysis like multiple sequence alignment to understand mutational changes in the genome of SARS-CoV-2 compared to different strains, as I have already told, strains from humans and also strains from different animals. Next, we will also see what are the important protein components of the virus through which they enter into the human cell. And we will also learn how to visualize the identified mutations on the spike protein, for example. And from such analysis and such visualization, we can understand and we can study which mutation might affect the functional properties of this spike protein and alter its ability to bind to the receptor in the host cell. So furthermore, we will also learn how we can look for transcriptomics data from patients that are infected with SARS-CoV-2 using public repositories like Gene Expression Omnibus database and etc. Here, we will explore such data, this data in particular, to find out important transcriptional signatures that are associated with drug treatments like ruxolitinib, Rux in short. We will also learn some important bioinformatics methods that are available for identifying gene expression signature for treatments like Rux or ruxolitinib. We will perform differential gene expression analysis between Rux treated and untreated samples. And we will also employ unsupervised machine learning methods like principal component analysis and clustering to understand the potential of these signatures in categorizing drug treated and untreated samples. So, during the course of this project, you will learn how to do and the logic behind such analysis and how to interpret these analysis in the context of the data or in the context of the biological project or question in hand. So next project we will get introduced to is on the outbreak of enterovirus D68 or EVD68 in short. This is a virus that causes a severe neurogenic disease called acute flaccid myelitis, AFM in short, mainly in children. So it is in comparison, it is a polio-like illness defined by the acute onset on spine or on the paralysis of the spine. So in this project, we will understand the evolutionary relationship between the strains of different outbreaks of this virus. So to go into the history, this virus first emerged around 1960s. And in the last decade itself, the outbreak of EVD68 has been reported across different parts of the globe, even in developed countries like United States, Europe, and roughly once in 2 years or once in 3 years or so on. So their pathogenicity also varies among these outbreaks and among different populations of this virus. So similar to the SARS project, here also we will use various phylogenetic tree reconstruction techniques and algorithms to find out whether the EVD68 strains from different outbreaks at different time points like 2014, 16, 18 are from the same virus or from the same strain or from the different strain or a related virus using next-gen sequencing data of the patients. So we will also see that phylogenetic analysis reflects the variations in the strain that causes these outbreaks in different years or in different time points. Furthermore, multiple sequence alignment will reveal a number of variations in the sequences of different viral strains. A careful analysis and how to perform this careful analysis will help us identify critical mutations that can cause significant changes in the amino acid level among these different strains. These mutations can be related to the virulent properties of the pathogen and will help us characterize the viruses like EVD68 or SARS-CoV-2, which is currently a very dangerous pandemic going on, based on their pathogenicity. Hope that was informative and that was very interesting. And I look forward to meet many of you in the Bioinformatics for Infectious